In order for the cell to enter various stages of the cell cycle, including the S phase and mitosis, CDK and cyclin activity must reach a certain level. Positive growth factors interacting with the cell triggers an increase in CDK and cyclin activity to promote DNA synthesis or mitosis. A growth factor is secreted by another cell. For example, if you get a cut on your skin, the skin cells around the cut will send a growth factor to other skin cells to begin reproduction. That growth factor binds to a receptor tyrosine kinase, causing the kinases to come together. This reception triggers the start of a signal transduction pathway. Kinases become partially activated. Then, a phosphate from 6 ADP molecules is transferred to the tyrosine amino acids on the receptors. Now, the receptors are fully activated. Inactive relay proteins then bind to the receptors. These relay proteins are now activated and amplify the original signal from the growth factor to stimulate cyclin CDK activity. This stimulation of CDK cyclin activity promotes stages of the cell cycle, including DNA synthesis, and in this example, mitosis, in order for the skin cells to repair the damaged cut. Helicase is an enzyme that breaks apart the hydrogen bonds between the nitrogenous bases of two DNA strands, splitting DNA into two separate parent strands.
The second step of DNA replication is when primase creates sequences made of RNA, which serves as a placeholder for DNA polymerase to latch onto. This RNA chain is a primer and synthesized by the enzyme primase. Primase starts as a complementary RNA chain with a single RNA nucleotide and adds RNA nucleotides one at a time, using the parental DNA strand as a template. The new DNA strand will start from the 3 end of the RNA primer.
DNA polymerase begins at the RNA primers and attaches complementary nucleotides to the nucleotides of the original parent strand. Polymerase reads in a 3 to 5 prime direction on the parent strand, but it builds the new daughter strand in a 5 to 3 prime direction. The polymerase building in the direction opposite of the helicase forms a lagging strand. The polymerase building in the same direction as the helicase forms the leading strand. More polymerase will appear on the lagging strand to continue to build the daughter strand as helicase unzips more original parent DNA. Polymerase also takes out the RNA primer and replaces it with complementary nucleotides.
The last step of DNA replication is when ligase connects any ends of DNA fragments that aren't connected. DNA ligase joins the sugar phosphate backbones of all the Ozaki fragments into a continuous DNA strand.
After DNA replication, there are now 2 identical copies of DNA that skin cells can use to produce more identical skin cells for repair.
Prophase is the first stage of mitosis, in which the chromatin condenses into chromosomes that are visible, the mitotic spindle fibers begin to form, the nucleolus disappears, and the nuclear membrane starts to break down. Metaphase is the second stage of mitosis, in which the spindle fibers attach to the kinetochores of the chromosomes and align them in the middle of the cell.
In anaphase, the proteins inside the centromere holding the sister chromatids together are inactivated. Then the spindle fibers pull apart the chromatids to opposite ends of the cell. Anaphase must occur so that each new cell has an identical number and copies of chromosomes. In telophase, chromosomes are at opposite ends of the cell. New nuclei begin to form around the chromatids. In cytokinesis, the skin cell finally begins to stretch apart at each end. The cell membrane finally splits, forming 2 identical daughter skin cells. The process of mitosis will continue until the cut is fully repaired by identical daughter cells.